Hello, good morning students. So today we are going to start with the cell membrane extensions. Though we have already started with the extensions of plasma membrane in prokaryotes. If you remember, yesterday we have talked about prokaryotic cells where we have talked about the nucleoid, which was the non-membrane bound nucleus found in prokaryotes. Apart from that, it has cell membrane extensions in the form of, the very first was mesosome, where it has role in cell division. What else we have discussed? In DNA replication. Also, we have talked it play role in increasing the surface area for enzymatic activity, if you remember, right? So, increase surface area for enzymatic activity. Though up till here we have already done in our yesterday's lecture. So today I am going to start with the second extension of plasma membrane, and that is what? Chromatophores. What is that? Chromatophores. So chroma means colored. Chroma means colored, and phores means body. So the colored pigment containing bodies found in the extension of plasma membrane is called as chromatophores, right? And is also exclusively present in case of prokaryotes, especially cyanobacteria. Is that clear? So let us see. Chromatophores are the colored pigment containing bodies found in prokaryotes, that is bacteria only, but not all the prokaryotes. So some bacteria such as the cyanobacteria or the blue-green algae. Is that clear? Yes? Okay. How do they look like? They look like this. Then having certain pigments in them. These are what? Chromatophores containing colored pigments, or you can also mention photosynthetic pigments found in prokaryotes such as cyanobacteria or blue-green algae. Is that clear? The third extension is Pili. Pili.
Hello.
Hello students, so let us resume our class again. Okay, so we have already talked about mesosomes, if you remember, and rest we are going to do now about other cell membrane extensions. Yeah. So mesosomes, chromatophores, Pili, fimbriae and flagella. So these are the cell membrane extensions that we need to know, out of which mesosomes we are already done, chromatophores we are already done. Now we are left with Pili, fimbriae and flagella. Mesosomes are the cytoplasmic extensions which play a role in cell division, DNA replication, cell growth, increasing surface area for enzymatic activity and all that we have already discussed yesterday also, and I think in today's class also. When we started, I started with mesosomes only. Okay, in chromatophores, these are the extensions of plasma membrane which stores what? Colored pigments. Remember, colored pigments. As I told you, chroma means colored, phores means bodies. So the bodies which stored colored pigments are called as chromatophores and are found in autotrophic prokaryotes. That means the cyanobacteria, which are also called as the blue-green algae. BGA stands for blue-green algae. Is that clear? Now next was another extension of plasma membrane which helps in mating. It helps in mating. Okay, it simply helps in mating, that is exchange of genetic material between the two prokaryotic cells or the bacteria, as I have shown you. But I think the lecture has stopped that time, that's why you guys were not able to see it. So this is simply exchange of genetic material. Is that clear? Yes. Next is the fimbriae, another extension of plasma membrane, which are bristle-like structure, which helps, okay, which helps in attachment with the host. What does it help? Attachment with host or the substratum. Okay, play role in. This was mating or the exchange of genetic material. Here it helps in attachment with host, or it can be with substratum. Means अगर बैक्टीरिया को किसी भी बॉडी से, be it other living organism or some non-living organism, it wants to get attached with something, that it has the extensions called as what? Fimbriae, which look like this. See, if this is the bacteria, then it will have hair-like outgrowths called as what? Fimbriae. Okay. Then there is Pili. Pili we are already done, which helps in mating. These are just one, usually one only. Then next is flagella. Flagella is the elongated structure made up of plasma membrane extensions only, which has role in simply motility, that is movement of the bacterial cell. Motility or the movement of the bacterial cell. This is simply flagella. Is that clear? So these are about the extensions of cell membrane which play a role in performing various activities in case of bacteria. Is that clear? I hope you all are clear with the concept. Just note it down and then we'll proceed with a very important concept or with a very important structure that is exclusively found in prokaryotes, that is plasmid. Yeah. So the next is plasmids. Let us understand what are plasmid. These are not the plasma membrane extension. It is a separate thing exclusively found in prokaryotes, only in prokaryotes. Okay? Yeah. Let us understand what is plasmid. See, the bacteria have the genetic material in the form of naked structure, like it is naked present in the cytoplasm. We have already discussed in yesterday's class, and that genetic material is called as what? Nucleoid or the incipient nucleus. Yes, we have discussed yesterday, nucleoid or the incipient nucleus. Right, bache? Now, you will wonder to know that the bacteria have, apart from the genetic material, they have one more structure called as what? Plasmid. And what is that? What is that? That is the plasmid, which is an extra chromosomal, double-stranded, DS stands for double-stranded, circular, self-replicating body called as what? Plasmid. So how does it look like? See, an extra chromosomal. Why do we call extra chromosomal? Because it is apart from this. Okay? See, here is the structure circular, then this is double-stranded, then self-replicating body found in the cytoplasm of prokaryotes only, called as what? The plasmid. Is that clear? An extra chromosomal, double-stranded, circular, self-replicating body is called as plasmid. Is that clear? And as I told you, it is exclusively found in prokaryotes. Is that clear? So the question is, ma'am, what is the role of this extra chromosomal DNA? If the genetic material is already present over here, then what is the role of this extra chromosomal DNA? So my dear student, this extra chromosomal DNA provides an extra feature, an additional feature to the bacteria. Let's say, like all the human beings, or me or you cannot sing like Lata Mangeshkar, right? Yeah, we cannot dance like Michael Jackson, right? So they are, gifted. They are God gifted with that particular property. Likewise, some bacteria are provided with an additional feature that is not present in all. So that additional feature is called as a unique phenotype. So this plasmid actually imparts an unique phenotype to the bacteria. And remember, this chromosome is apart from the genetic material. That's why extra chromosomal, circular, self-replicating means it is autonomous, independent of other. So that is all about the plasmid. You can note it down, and then we can proceed further with the ribosomes. Yes, noted. Plasmids are extra chromosomal DNA exclusively found in the prokaryotes, right? That's all about plasmid. Now one more structure that is found in the cytoplasm of prokaryotes, that is the ribosomes. Now you will ask me, ma'am, but ribosomes are also found in eukaryotes also. So yes, my dear, basically ribosomes are the protein factories. So they are found in both prokaryotes as well as eukaryotes. But there is a difference between eukaryotic ribosomes, prokaryotic ribosomes, that I will discuss now. Okay? Moving ahead, the next structures that is found in case of the prokaryote is ribosome. And what is the function of ribosome? As we know, The function of ribosome is to synthesize protein. That's why they are called as protein factory. Okay, ribosomes are called as protein factory, found in prokaryotes and eukaryotes both. So what is the difference between the eukaryotic and prokaryotic protein factory? See, the prokaryotic ribosome is of 70S type. What does this 70S type means? Means it has two subunits. The larger one is the 50S and the smaller one is the 30S. So the question is, ma'am, is this 70S also found in eukaryotes? No. The eukaryotic ribosome is of, let us discuss this point separately because we are now discussing about prokaryotes. So in prokaryotes we have 70S type of ribosomes. We have 70S type of ribosome in prokaryotes. Okay. While in eukaryotes you will have 80S. You can write down it separately. And there the units are 60S and 40S with two subunits, the larger 60 and the smaller 40S. Is that clear? But this is for eukaryotes, whereas this is for prokaryotes, whereas the S stands for the name of the discoverer, that is Swedberg's coefficient, or sometimes we also call it as the sedimentation coefficient. Is that clear? Yeah, because the units of the ribosome are separated on the basis of sedimentation. That's why it is also called as sedimentation coefficient. Is that clear? So that is all about the ribosomes found in the prokaryotes and are involved in protein building. That's why protein factory. Now if the ribosome has to synthesize proteins, now they are entering the process of protein. So what happens? Protein synthesis के time पर these ribosomes get united. These ribosomes get united on a thread-like structure that is called as mRNA. So on this mRNA, the thread-like structure get united and the ribosomes are attached with this, like this form. Multiple ribosomes will be united on the thread-like structure. Now this multiple ribosomes on this thread-like structure is given a special name that is called as polysome. That is called as polysome or polyribosome. Or there is one more name, and that is ergasome. Ergasome, polyribosome, polysome, all is the name given to this multiple ribosome attached with the mRNA called as polyribosome, which is formed during the process of protein synthesis. Is that clear? So I hope you all are clear with this. Just note it down as soon as possible. We'll proceed further. Yes. If there's any doubt, you can ask me. Like in tomorrow's class, you can ask. Okay. Yeah. Shall I proceed further? Last but not the least about your prokaryotic cell is the inclusion bodies. Now we are moving towards the inclusion bodies. So what are inclusion bodies, dear students? See, till now we have not seen any structure in the prokaryote which is membrane bound. Like all the structure, be it ribosomes, nucleus, cytoplasm, like none of the structure which is found in the prokaryotes is membrane bound. Only the boundary is there, that is in the form of glycocalyx cell wall and the plasmembrane. Otherwise ribosome, nucleus, none of the structure is with cell membrane. So how can this inclusion bodies which is also found in prokaryotes can be cell membrane? So this is a non-membrane bound structure found in prokaryotes which play role in storage or act as reservoir. Okay, it acts as reservoir or act as storage body, right? It acts as storage body. Storage of what? It can be cyanophyscian granules. Okay, these are type of certain pigments, cyanophyscian granules. It can store some ions like phosphate granules, etc. So these things can be stored, or also they can store air. So we can also say they can act as air or simply gas vacuoles. They can act as gas vacuoles, which helps in maintaining buoyancy. That is simply helps in floating of the prokaryotes. Is that clear? So inclusion bodies are also found in the prokaryotes which play role in reservoir or act as storage body, can store cyanophyscian granules, phosphate granules, and can act as maintaining buoyancy. And note it down, this is very, very important for your asked so many times. So that is all about your prokaryotic cell system. Is that clear? That is role in floating. Okay, so that's all about the prokaryotic system. Now let us move towards the another topic, that is the eukaryotic system. Is that clear? Yeah. Yeah. So U means true and karyot means the nucleus. Okay, so the one with the true type of nucleus bearing cell is simply called as eukaryotes. And not only true nucleus bearing system, rather also the cell which bearing all the membrane bound organelle is also placed in eukaryotes. Is that clear? Yes. So what are the salient features if we talk about eukaryotes? Then it consists of, it has a well membrane bound nucleus. Okay. Then if we see, it has well membrane bound organelle, right, such as ER, Golgi, lysosomes, vacuole, etc. is there. Okay. Then we see it has ATS type of ribosomes are also there in case of eukaryotes. Then in eukaryotes we will see a proper division of labor. Means, as we saw that in a bacterial cell there was just a cell having its genetic material and all the activity was performed by the plasma membrane or the cytoplasm itself. But here you will see, if I draw a plant cell, then you will be able to see that we have different organelle meant for different function. So can I say there is division of labor, proper division of labor seen in eukaryotes. Division of labor is present. For example, if there is a factory, okay, there is a factory, then one of the cell is performing its refining of the raw material. Then here there is manufacturing. Then here there is, you need not to draw this diagram or this part, I am just giving you for the explanation, like refining of raw material, manufacturing, then there is packaging, then there is tagging, and then there is commercialization and going to it for sell. Okay, so we can say all the different activities are not performed by a single cell, rather we have separate segments for performing different activities. Likewise we will see here we have different structure for performing different activities, like this, your nucleus, okay, followed by nucleus we have the ER, okay, we have the ER, right, then followed by the Golgi bodies, we have Golgi bodies, then we have lysosomes, okay, then we have the mitochondria, then we have the large size vacuole is also there, okay, yeah, then we have peroxisomes, then we have chloroplasts, very important, we have chloroplast, right, then we have outermost cell membrane and the inner is outermost cell wall and the inner is cell membrane is also present, right, and then we'll also see between two adjacent cells, if there is passage of molecules between two cells, like this, you can say there is passage of molecule between two cells in a plant cell, in a B plant cell, there is passage of molecule like this, the molecules can go from one side to another side, then such passage of molecules found in the plant cell is called as plasmodesmata. But we will be discussing about plasmodesmata later on, but now I just wanted to label all these organs. These are what? Lysosomes. Okay. These are what? Chloroplasts. Green color are chloroplast. Okay. These are what? Golgi. bodies. Okay, these are what? Endoplasmic reticulum. Okay, this is what? Nucleus. Fine. This is what? Vacuole. Okay, then this is what? The mitochondria. Let us draw it more precisely. This is mitochondria. This is mitochondria. Yeah, very sausage shaped structure is mitochondria. Yes. And the outermost yellow layer is the cell wall, and the second layer is green color is the cell membrane. Is that clear? Now you can draw it, you can copy it, and you can see if there is any doubt, you can just ask me. Right? Yeah. And you must be aware of this thing. If I am erasing this plasmodesmata as if now, we will study it once again. See, if on the ribosomes, if on the endoplasmic reticulum, if the ribosomes are present, you can see. If the ribosomes are present on the surface, then we call them rough endoplasmic reticulum, and if the ribosomes are not present on the endoplasmic reticulum, then we simply call as smooth endoplasmic reticulum. Means endoplasmic reticulum is of two types, SER and RER. If ribosomes are present on the surface like this, then it is called as rough endoplasmic reticulum. If it is smooth like this or this, okay, then it is smooth endoplasmic reticulum. I hope I am clear with the concept. You can just see, you can just note it down, and we'll proceed further with the components of eukaryotic system, that is the cell wall, cell membrane and all that. Okay? Yeah. Proper division of labor is seen here in case of eukaryotic system. Now we will start with the outermost covering, that is cell membrane in case of animal cells and cell wall in case of plant cell. Right? So let us start with the outermost covering, that is the, if I talk about the cell envelope, if I talk about the cell envelope in case of eukaryotic cell, then it consists of two structures, cell wall and cell membrane. Okay? This is only found in animals. Okay? Whereas together is found in plants. Okay? Like plants have cell wall and cell membrane both. The outermost covering is cell wall. The cell membrane is exclusively found in animals. Okay? So first I am going to discuss with this non-living rigid structure which plays role in the protection, providing a definite shape. We have discussed yesterday also. It prevents the cell from the bursting. Is that clear? So let us talk about the cell wall. Okay? Cell wall. See, the cell wall, if this is a cell, okay, cell wall in the plant cell is the outermost covering, and you must be aware of this thing. You must be aware of this thing, that it is, cell wall is a non-living. It is a non-living rigid structure, okay, that is made up of cellulose and pectin, in case of plants. But if it is algae If it is algae, it has additional galactans and mannans. Okay. If it is chitin, then it has different composition, and that is the fungi. And if it is bacteria, we have already discussed yesterday what it is made up of. I hope you guys will recall the composition of bacteria. So you will be writing this here by your own. I am giving you here as homework. Here as homework. So you can see the rigid structure, the non-living structure, cell wall is made up of different substance in different organism. Is that clear? If it is the plants, it is made up of cellulose and pectin. Okay, cellulose, pectin, and there is also hemicellulose. And if it is bacteria, I have discussed yesterday. Okay, in case of algae, there is galactans and mannans. In plants, we also have hemicellulose. Okay, so that is about the composition of cell wall in case of different organism. Now one more thing you need to be aware here about the cell wall, that the first formed cell wall, the initially formed cell wall, is simply called as what? The primary cell wall. The primary cell wall, because we do first primary schooling. So the first cell wall formed is primary, and the one which is formed later is what? Secondary cell wall. And the same cell wall, primary cell wall, get deposited with the one more secretion and converting the primary cell wall into secondary cell wall. You must be aware of this thing. Primary cell wall is capable of growth. But once the secondary cell wall is formed, the growth stops. It becomes thick, it becomes dead, and the growth stops. Okay, so that's all about the non-living nature, rigid structure, cellulose, pectin and hemicellulose in plants, galactans, mannans, algae, fungi, chitin and bacteria we already discussed. Now we'll be talking about the function of cell wall. Is that clear? Yeah, and one more thing. The secondary cell wall will be outside or inside? Secondary cell wall will be always formed from inside. So it will be formed inner to primary cell wall, because always the cell is making some of the components from inside only. Okay, and then making the primary cell wall getting converted into the secondary cell wall. So the first formed is primary, the second formed is secondary. Primary is capable of growth, secondary is not capable of growth. I hope I am clear with the concept. Moving ahead, the functions of cell wall, till that time you can just go through. Non-living, rigid structure. For plants, cellulose, pectins, hemicellulose. For algae, galactans and mannans. Though it is in more detail when we will be talking about the animal kingdom, the plant kingdom, but here this much is sufficient. Chitin, fungi, bacteria, it was peptidoglycans and lipopolysaccharides. Okay, that's all about. Then the functions we have already discussed in our yesterday's class. That was protection, providing definite shape, and the third is your prevents from bursting. Is that clear? That's all about the function of cell wall. Next I am going to start with the cell membrane. Yeah, so be ready for the cell membrane, which is the second structure. Now cell membrane. Cell membrane in eukaryotic cell is made up of lipids and proteins. Okay? Yes? How much lipids and how much proteins? Yes? Have you gone through your NCERT? So, 40% is your lipids and 52% is proteins. Then definitely one more component would be there that we will be discussing. Actually, your plasma membrane also consists of some sort of sugar also, but we don't mention it over here because in NCERT they have just given 40% of lipids, 52% of proteins and some percentages of sugar as well. Okay, but before that, who has discovered the structure of cell membrane? And it was given by Singer and Nicholson after the advancement of microscope, your electron microscope in 1972. So the first time the proper structure of cell membrane was given by Sanger and Nicholson in 1972 on RBC, on human RBCs. Singer and Nicholson has given the structure and that model was called as fluid mosaic model. Is that clear? That model is called as fluid mosaic model. Now how does it look like? What is that? Also it is called as phospholipid bilayer. So I am going to make you understand how is the plasma membrane look like. Cell membrane consists of lipids and proteins. Then it consists of, basically it was discovered by Sanger and Nicholson. Then it is also called as fluid mosaic model and it is made up of phospholipid bilayer. That is why it is called as phospholipid bilayer. Yeah. So let us draw the diagram of this phospholipid bilayer. Okay. Cell membrane consists of lipids and proteins. Okay. This is basically not Sanger, this is Singer. We call it as Sanger, but this is written as Singer and Nicholson. Okay, yeah. So let us draw the diagram and see the structure of plasma membrane. See, I have written the phospholipid bilayer. I have written those phospholipid bilayer. So let us draw the phospholipid bilayer. Okay, yeah, like this. See, what I am drawing over here, I am drawing the hydrocarbon chain which will be attached with the phosphate group. Let us draw the phosphate group over here. These are the phosphate group, Every tail will have phosphate group. Okay, phosphate group and phosphate group. Right. So this is the phosphate group. Okay, attached with the, this is what? Hydrocarbon tail. This is
hydrocarbon tail, and it is together making what? Together making one lipid. This is what? A lipid molecule. This is making what? A lipid molecule. So my dear student, one lipid molecule, one lipid molecule, one lipid molecule, one lipid molecule, and like many lipid molecule unite to form what? One lipid layer, one lipid leaflet. Okay, so many of the lipid molecules combine to make one leaflet, and the another molecule layer is making another leaflet. So it is together called as phospholipid, because this is phosphate containing lipid, so it is called as phospholipid bilayer. Is that clear? Why do we call it as, in our previous slide we just saw, it is called as phospholipid bilayer. Why phospholipid bilayer? Because one lipid, as I can draw over here, one lipid is having two hydrocarbon chain and one phosphate. This is the phosphate group which is negatively charged, and this is the hydrocarbon chain, okay, together called as phospholipid. Okay, and this chain is hydrophobic in nature. How is that? Hydrophobic in nature. That is, we can call it as non-polar. It do not react with water. Okay, it do not react with water. It do not come in vicinity with water. It actually hates water. That is why hydrophobic. Okay, and the one which loves water is the phosphate group, because it is charged, so we can label it as hydrophilic. Is that clear? Hydrophilic end. Okay, hydrophilic end, that can attract water or that can react with water. Is that clear? So the same molecule is having hydrophilic end, and the same molecule is having hydrophobic end. So together we can call this phospholipid molecule as amphipathic molecule. Is that clear? And because it is having two leaflets, we call it as phospholipid bilayer. Is that clear? And in this phospholipid bilayer, we said it is made up of 40% lipids and 52% proteins. So where are the proteins, my dear students? Where are the proteins? They are not visible here. So let us draw here proteins. They are actually embedded within the membrane. You can see like this. They are actually embedded in the membrane. They are actually embedded like this in the membrane, like this, like this, like this. Okay, we will see it has two type of proteins. One, the bigger size of proteins which are crossing entire membrane, called as integral proteins, and the one which are located at periphery are called as the peripheral proteins. So this was about the lipid. Now we are talking about the proteins here. Proteins. So we have two type of protein. One is the integral proteins. Another is the peripheral proteins. Okay, the one cross entire membrane is called as integral proteins, like the bigger red color proteins like this. Okay, and the one which are located at peripheries like this are called as peripheral proteins, are present at periphery, are called as peripheral proteins. Is that clear? So the peripheral proteins are easily released from the membrane if the membrane is agitated, right? अगर आप membrane को तेजी से हिलाते हैं, so the peripheral proteins can come out, but the integral proteins do not come out because they are embedded, they are deeply buried into the membrane, right? So that is called as integral protein. Is that clear? Now we have given the name of fluid mosaic nature. Okay, fluid mosaic nature or the quasi fluid nature. There is one more term for the plasma membrane. We say that it shows the quasi fluid nature. Quasi fluid nature. So what does it mean? See, actually your plasma membrane look like this. Sea of lipids where proteins are dispersed in it and floating like icebergs. Okay, on the bed of lipids proteins are floating like icebergs. So the plasma membrane, can you say plasma membrane is a solid structure? No, we cannot say it's a solid structure, because if it will be a solid structure, it will not allow the passage of molecules across it. So what do you say? Plasma membrane is a liquid structure. Yes, if it will be liquid structure, then it will not be present at a particular point, it will flow away, right? So what is that? Actually, plasma membrane is neither solid, it is nor liquid, it is semi-liquid. It is semi-fluid. Okay, semi-liquid or semi-fluid. That is why we call it as the fluid mosaic model or the quasi-fluid. That is, it is semi-fluid matrix like membrane is there, semi-fluid covering is there around the cytoplasm, making what? Plasma membrane, and we call it as fluid mosaic model or the quasi-fluid nature. Is that clear? Yeah. Now you can note it down. If there is any doubt, you can ask me. If not today, you can ask tomorrow. See, here, what do you think? What is moving? The proteins are moving or the lipids are moving? Here, the lipids are floating in the membrane itself. And that floating movement of the lipid is responsible for the fluidity of the plasma membrane, the fluid nature of membrane. So we can write, we can add it here a point that fluidity of membrane. Okay, what do we write? Lipids are responsible for fluidity and elasticity of membrane. That means the lipids are the substance in the plasma membrane which is providing the fluid nature or the fluidity of the membrane or the flexibility or the elasticity of the membrane, over which the proteins can also move and also the molecules can also move across the membrane. Okay? Be it via transmembrane proteins, the integral proteins, if the molecules are polar, then they will move through the integral proteins, and if the molecules are non-polar, then they will move through the lipids. But lipids are responsible for the fluidity and elasticity of the membrane. That's all about your fluid mosaic model or quasi-fluid nature given by Singer and Nicholson, which was given by which was given by Singer Nicholson in 1972 and on the RBCs. That's all about now the functions of plasma membrane. Next is functions. So mainly it is involved in the transport of molecules. It is involved in the transport of molecules. Now the transport can be of two types. It can be active or it can be passive. Okay, if the molecule is transported using ATP or expenditure of energy, then the transport is active. And if the molecules are transported without expenditure of energy, that is ATP, then we call it as passive. So transport of molecules across the plasma membrane can take place in two ways. One is the active transport, another is passive. If it is active, it is by using ATP. If it is passive, it is without any expenditure of energy. One more thing. So ma'am, when do we need ATP and when we don't need ATP? See, if I am having 1000 rupees, then I can easily pay you 500 rupees. But if I am already having 500 and I have to pay 1000, I am having 500 and I have to pay 1000, then what will happen? It will require energy. Either I will borrow from somebody or I will earn from somewhere and then I can pay you 1000, because I am having 500, you are asking for more. My concentration is less, I have to give somebody who needs more than 500. So I am having low, you are demanding 1000, so I need to spend energy. So when from low concentration to high concentration, then it requires energy. From low concentration to high concentration, then it requires what? Energy. Whereas in passive, from high concentration to low concentration, then it is passive. Is that clear? And I hope you guys have also recalled that fluid nature of the plasma membrane is due to the movement of lipids, not the proteins. Is that clear? So that's all about the function of your plasma membrane as well. Okay? It allows the molecules selectively to enter inside and selective to leave out. That is why plasma membrane is also called as selectively permeable membrane. Please note it down, very, very important. The plasma membrane is a structure which we call it as selectively permeable membrane. That's all about the plasma membrane of eukaryotes. Is that clear? Yeah, now we will be moving towards the other organelle. Though we have done about the cell wall, we have done about the plasma membrane also. Okay. Now some important organelle such as chloroplast and mitochondria, etc. Okay? Yes. So let us start with the chloroplast. Number 1. One by one we will be moving towards the organelle. Chloroplast, which is exclusively found in plants. So till now we have saw two structure which is only found in plant. One was the cell wall, second is the chloroplast, which is commonly called as what? Kitchen. है ना? Kitchen of the cell. The place where the plants synthesize their food is called as kitchen. Okay? So the question is, what is there present in the chloroplast that it can synthesize their food? Why not we can also synthesize our food? So now you will say, ma'am, hum bhi to khana bana lete hain. So it is not the khana, it is not the actual khana. Basically, You are just preparing a already prepared food. Like we have prepared starch, we have prepared vegetables, starch is there, potato, rice, wheat, all is already prepared in the form of starch. Now you are just boiling it, you are just cooking it. But can we directly synthesize starch? No. So why no? Because we don't have chloroplast, we don't have the photosynthetic pigments in them which can capture solar radiation and it can convert them in the form of the glucose, that is CO2, C6H12O6, which is formed by combination of CO2 plus H2O in the presence of solar radiation, and the solar radiation is captured by the pigments present in the chloroplast. Is that clear? So chloroplast is the kitchen of the cell or the site where photosynthesis takes place. Let us draw once again. If this is the leaf, okay, if this is the leaf, then it has special cells called mesophyll, which consist of the chloroplast, and that chloroplast consists of chlorophyll pigment, okay, which capture sunlight. Okay, so let us see the structure of chloroplast over here. Okay, chloroplast is a double membrane bound structure like this, which has membranous extension present inside like this, like this. The membranous structure present inside is called as grana, if it is in the form of stacks like this, and if it is present dispersed, then the individual units, these individual units is called as thylakoid. The membranous structure, individual units are called thylakoid, and the entire pile of the thylakoid is called as grana. If it is plural, that is grana. If it is singular, that is granum. Granum is singular. Singular, this is what? Plural. Okay? So the membranous extension present in the chloroplast is called as lamellae, is simply called as the lamellae. If it is making this individual units, this lamella is called as thylakoid. Thylakoid, different, multiple thylakoids making a stack, called as grana. Okay. Now multiple granas are interconnected with the help of filamentous ribbon-like structure called as stroma lamellae. Is that clear? Stroma lamellae. This is outer membrane. This is outer membrane. This is inner membrane. What else is present in the chloroplast? Because chloroplast is synthesizing sugar which is stored in the form of starch. So we will be drawing what? Starch droplets. These are the food. These are starch droplets. And my dear students, you will wonder to know that the chloroplast also consists of the ribosomes, which was present in prokaryotes, which is also present in eukaryotes, as well as it is also found inside chloroplast, because chloroplasts need its own proteins to be synthesized. So the chloroplasts have its own site of synthesis of proteins, and that is in the form of ribosomes. So let us draw the 70S type of ribosome also is present over here. Okay. What else? What else? They have their own circular DNA. So can I say the chloroplast have its own ribosome, has its own DNA, and has its own food in the form of starch droplets. Is that clear? So my dear students, if the chloroplast is having its own DNA, if it is having its own ribosome, it is having its own food, so don't you think it can run independent of any other regulatory system. Means it is not directly regulated by nucleus, it has its self-regulatory mechanism, and that is why this is called as autonomously replicating structure like that of the plasmid in prokaryotes. Here we can call it for chloroplast, and in upcoming lecture we will see we can also use the statement for mitochondria. Is that clear? What did I say? It is an autonomous regulated body for chloroplast. So we can write here autonomous regulating or regulated body. Chloroplast can replicate by itself. It is regulated by itself. Now you can note it down what is unique about the chloroplast and what it consists of. Is that clear? Yes. And one more thing. The vacant space that you see here, this all vacant space that you see here is actually called as stroma. Very, very important. You need to recall it. What is stroma and what is the thylakoid or the grana part. Okay. We will discuss while studying the photosynthesis. Okay. Yes. Now the question is, ma'am, you said in chloroplast the pigments are present. In chloroplast, like mesophyll, chloroplast, the chlorophyll pigments. Where these chlorophyll pigments are present? So the chlorophyll pigments are present in the thylakoid. Okay. Chlorophyll pigment is present in the thylakoid, and thylakoid also have mechanism to synthesize ATP. It has mechanism for synthesis of ATP similar to that of mitochondria. See, mitochondria is the powerhouse of cell in animals and plants both, but in plants there is an additional site where ATP can be synthesized, and that is the thylakoids of chloroplast. So that is why I have written it separately, that they also have mechanism to synthesize ATP similar to that of mitochondria. Okay, that's all about the chloroplast. Now we can proceed further. We will be sharing the slide to you guys so that you can note it or add in your notes. Now next is the type of chloroplast. Depending on the type, we have various type of chloroplast, or we can say plastids. Actually, this chloroplast is also a type of plastid only, right? So apart from chloroplast, chloroplast is the one which consists of chlorophyll pigment. Now we have two more types of such structure like chloroplast. Another is leucoplast and the chromoplast. See, this chloroplast was actually chloroplast comes in category of plastid. What is this? Plastid. What is this? Plastid. Plastid. Actually, chloroplast is a type of plastid. These are the structure which consist of several type of pigments or meant for storage. Plastid, TID. So one was the chloroplast, which we have already done. Okay, which consists of green color pigment. Right? Another is second type, that is what? Chromoplast. Chromoplast, which consists of several other color pigments which are responsible for the colored pigments of flowers like red, blue, orange, etc. So that is what chromoplast is consist of. Have different colored pigments. Have what? Different colored pigments, which is like we can consider it similar to pigment containing plastids. This is also pigment containing, but this is the green color pigment containing, that is why it is important, and that was actually chloroplasts. Apart from green color pigments, else are present in the chromoplasts, but the most important type of chloroplast that we need to know here is the leucoplast. Very, very important. Leucoplasts are the colorless plastids. These do not have any pigment. These are colorless plastid which are meant for storage. Of what? Nutrients. Storage of nutrients. One is the aleuroplast. Another is the elaioplast, and last is the amyloplast. Basically, these are the leucoplast. Leucoplast is further type of plastid. See, the general category is plastids, which is of three types: chloroplast containing green color pigment, other color pigments are chromoplast, and no pigment is leucoplast. So what are for leucoplasts are meant for what? For storage of nutrients. It consists of aleuroplast for storage of proteins, elaioplast for storage of fat and oils, and amyloplast are meant for storage of what? What is left here? Carbohydrate. Is that clear? So that's all about today's class. Rest we will be discussing tomorrow. Alright. So chloroplast is what? We can give a quick recap. Chloroplast is a kitchen of the cell, autonomous regulated body. Why do we call it autonomous? Because it consists of the ribosome, has its own DNA, has its own starch synthesis particles, has its own site for ATP synthesis. That is why it is autonomous regulated. It is a double membrane bound organelle. You can see outer membrane, inner membrane. Then the inner vacant space is called stroma. Then these units are called as, single unit is called as thylakoids. The entire stack is called as grana. And the interconnection between the grana is called as stroma lamellae, which is the site for containing pigments. Basically, pigments are present where? Chlorophyll pigments are located in thylakoid membrane. Alright? And this was about the type of plastid. Basically, chloroplast is also a type of plastid having green color pigments, having different pigments, chromoplast, no pigment, chloroplast, site of storage of different nutrients, aleuroplast for proteins, elaioplast for fats, and carbs are stored in amyloplast. That's all about today. Thank you. Have a good day.